Thanks everyone for coming. I'm extremely excited to get to share kind of my nerd hat with all of you. And I'm relatively new to the sustainability school and the sustainability world, but it's been really fun over the last year and a half to get to meet all these wonderful new scientists and hear about everybody's ideas for how we can build a better and more sustainable future. And often these ideas have to do with big things like changing energy policy or coming up with new sustainable food sources. And hopefully today what I will do is plant in your ideas, in your head, the idea that there may be some other solutions that are a little bit less obvious, but that are beneath our feet all the time as we're walking around in the world. And to start, I want to begin talking about mushrooms. This is my entree into thinking about subterranean sustainability solutions. And if any one of us was to take an afternoon off during the rainy season and go for a hike around one of our California forests, if you're like me and you spend time kind of looking down and poking around, you can come up with this vast kaleidoscope of mushrooms here with their varied colors and varied shapes and sizes. And it's really in many ways a non-rational love of these organisms and their aesthetics and their ephemeral nature that really hooked me on the research that I'm doing right now. And in a really simplified way, this is what I study if you don't know me. I study mushrooms and I study their diversity and how this diversity affects the world around us, right? But I am fascinated by mushrooms in part because they're beautiful in their own right, but also because mushrooms are a signpost to something more amazing that's going on below ground. And this is because, there should be no volume here. And this is because mushrooms are really the tip of what you might think of as a fungal iceberg. They are like apples on an apple tree. And if you were to go to a mushroom and look beneath it, you'd see these microscopic filaments called hyphae that emanate out from beneath the mushroom. And this is sometimes a hard thing to imagine, so I'm just showing you a really brief part of a clip from a movie called Habitat Earth that plays in the California Academy of Sciences planetarium. If you haven't seen it, I highly recommend going to check it out. And I do it because it gives you this really amazing look, even though it's a little bit dark here, into this subterranean world where recently it was estimated that 60% of all species on the planet reside. And collectively, these hyphae form a network that's known as a mycelium. And this mycelium is really unique, this branched filamentous growth form that fungi have. And it's also really unique in forming this living tapestry that quite literally interconnects the different types of microorganisms and different abiotic environments that you find in the soils. All right, so we're going to end it there. But I really highly recommend going to the California Academy of Sciences and seeing this in the planetarium. But the most important part about the mycelium for this talk is that if you were able to follow this mycelium back, some of the mycelium from some of these mushrooms would connect back to the roots of a plant. And there they form this kind of intermingled plant and fungal tissue that's known as a mycorrhiza. And here they form this new symbiotic organ. And mycorrhiza literally means fungus root. And this might seem like somewhat of an unusual arrangement, but everywhere we've looked in nature, mycorrhizal symbiosis is really the rule rather than an exception. So it's been estimated that somewhere between 80 and 90% of all plant species form this type of a mycorrhizal interaction. And from some of the work we've done trying to map the global distribution of these kinds of symbioses, the abundance of plants forming mycorrhizal symbiosis is even more common when you think about the relative abundance of the individuals, the dominance of individuals. And so in one study, we've estimated that over 97% of all trees form some type of a mycorrhizal symbiosis. So this is really the way in which our terrestrial ecosystems work. So, why is this important? Particularly if we're thinking about sustainability. And this is because in this hybrid symbiotic organ, the mycorrhiza, the plants and the fungus are involved in this mutual economic exchange that's beneficial to both of them. So these fungal hyphae are foraging out through the soil, and they're scavenging things like nitrogen and phosphorus, and they're giving these back to the plant. And nitrogen and phosphorus are the two macronutrients that most often limit the productivity of terrestrial ecosystems. And so we all depend on plants and plant growth for building materials and food, and so this nutrient provision function of these mycorrhizas is really critical for the functioning of ecosystems and for human economy. These plants also receive a fairly large benefit. These plants get this large benefit. And in one study, it was estimated that somewhere close to 86% of all the plant nitrogen came from mycorrhizal symbiosis. So this is a fairly large flux. And there's a correspondingly large flux of energy from photosynthesis that goes below ground to this fungal mycelium. And so in one study that was recently published, they estimated that somewhere on the order of 13 gigatons of CO2 equivalents go below ground to these mycorrhizal fungi and their mycelium out in the soil. And to put this in context, this is about 36% of carbon dioxide emissions from fossil fuels. So this is a fairly large flux. So as you might imagine, given the magnitude of these carbon and nitrogen fluxes that I just described, any sort of an anthropogenic disturbance to these partnerships could have really negative consequences, like decreasing terrestrial productivity or carbon storage in soils. But for historical reasons, microbial organisms like fungi and bacteria have often been studied at very small spatial scales. And there's a disconnect between the scales at which we've studied them and the scales at which things like climate change are playing out. So until fairly recently, it's been very difficult to try and understand how these types of organisms might respond to climate change. So this is one of the things that my lab has been really focused on over the last decade. So we've been leveraging kind of advances in DNA sequencing, ecological theory, and big data analysis to try and understand what controls the distribution and abundance of these mycorrhizal fungi and their hosts, and how these symbioses function across large spatial scales. And while this might seem like a relatively esoteric pursuit, I think it's really only if we can understand the global drivers of these symbioses can we make predictions about what's going to happen in the future, what sort of feedbacks might develop, and if there are problems, how might we intercede, or more optimistically, how might we be able to leverage these partnerships to come up with novel sustainability practices. So just to give you kind of one example of some of the things we do, this map shows a large sampling network. And you can see these different points on here. These are places where there are sampling sites. All the places in pink are places that our lab group has gone and done our own kind of field sampling. And then we started this in 2011. And then the blue points are from something called the National Ecological Observatory Network. And each of these sites, you can see the little call-out boxes to the right. There are often multiple plots. And within each of these 40 by 40 meter plots there, you can see these little dots. And these are places where soil samples are removed for analysis. And when we started this project in 2011, it was really one of the largest scale soil sampling projects for microbial biodiversity around. And combined with what's happened since in NEON, we've had this very substantial database from which we can begin to build this large-scale understanding of fungal communities. So, from each of these samples, so we'll collect a soil sample. They don't always have these really charismatic mushrooms in them. In fact, they mostly do not. We take this, we'll do some chemical analyses of the soils. We'll then extract the DNA from these samples, use fungal-specific PCR primers to amplify marker genes that we use. We then use high-throughput sequencing to sequence all the DNA from these marker genes in our samples and use bioinformatics to identify the different fungal taxa that are present in them. And then ultimately, we'll take all of our information about local climatic conditions or soil environment and build statistical models that are going to allow us to then understand what are the key variables that predict the local diversity and composition of these fungi, and ultimately to begin to make extrapolations about what might happen in the future if conditions change. So from all of this sequencing, we're now starting to get a good handle on what mycorrhizal biodiversity looks like at a range of different scales. And so we now know that in a small handful of soil, in any sort of typical forest or other type of terrestrial ecosystem, there's often dozens of species of mycorrhizal fungi that are present. This, you can imagine how this scales up to the root system of an individual plant. So an individual plant can be simultaneously associating with dozens, if not hundreds, of species of mycorrhizal fungi that are very functionally distinct from each other. And these fungi are also embedded in an even more complex community of hundreds of other species of fungi and potentially thousands of species of bacteria. And this local scale richness and community structure scales up as you start moving across landscapes and out across continents, so that across North America there are thousands of species of these mycorrhizal fungi. And these fungal communities are regionally distinct from each other. So this map here shows one of the studies we did mapping the composition and diversity of fungal communities in pine forests across the sampling network. And this technique we've used here shows the dissimilarity of these fungal communities based on the colors here, right? And so you can see that similar colors are places that have similar communities of fungi. And so you can see that with these kind of red and yellow colors up in the boreal forest, those fungal communities are very distinct from the communities we see, say, down in the southeast that's dominated by these pinkish colors. And there's often very little overlap in the composition of these fungi from place to place. There's a lot of variability regionally. And our analyses show that climatic variables, things like temperature and precipitation, are often some of the strongest predictors of this diversity and of this regional variation. And this is somewhat novel in that people have often thought that the things that most strongly control the biodiversity of soil organisms are things like soil chemistry, pH, or soil carbon content. And one of the reasons we've worked so hard to develop these models is that it allows us to begin to scale up and make predictions about what might happen in the future. This is a figure showing our future predicted changes in the biodiversity of these local fungal communities by 2070 under this RCP 8.5 kind of business as usual emissions scenario. And what you can see is that we expect there to be fairly large decreases in the biodiversity locally of these symbiotic fungi. And this is represented by these warmer colors, represent larger losses of biodiversity. And this extrapolates out to median losses of 20% of species across an area the size of Alaska, so about 3.5 million kilometers square. And just as a side note, the magnitude of these losses in biodiversity change under different emissions scenarios, right? So this is something that human economic decisions will influence in the future. And these predicted losses in fungal biodiversity actually match up reasonably well with some other modeling we've done where we've tried to predict the dominance of different kinds of mycorrhizal symbiosis across forests globally. And these red colors, again, show large decreases in basal area. And this is for the particular type of mycorrhizal symbiosis that's most dominant in boreal forests. And again, you can see this matches up in the boreal forest there. We expect or predict large decreases in basal area of trees with this type of mycorrhizal symbiosis, somewhere on the order of 30 or 40% of basal area. Right. And we've done a number of other studies that show similar magnitude changes in these below-ground communities. So why does all of this matter? Well, I think together all of this shows that we really need to be thinking about the weather underground because it is really having strong impacts on these organisms, and they're critical components of the terrestrial biosphere. And we don't yet well understand what something like a loss of 20% of the local richness of these mycorrhizal fungi is going to do to the biosphere. All right, so these results should be concerning, but I want to try to end this talk with some optimism because our work shows the vulnerability of these partnerships to climate change, but they also indicate possible solutions, right? The glass half full of the work we've done with these molecular tools shows that there's an incredible local and regional diversity of these mycorrhizal fungi. And so our group and others, you can see indicated from some of these press clippings here, believe that there's a vast genetic potential out there that could be harnessed to try and build novel sustainability solutions. Things like creating more resilient plant communities or increasing below-ground carbon sequestration. So, the other branch of our research is going back into the lab to do experiments with these mycorrhizal partnerships. And for example, we've done things like expose plants and their mycorrhizal partners to drought stress and use transcriptomics to try and characterize the genes that are involved in response or acclimation to these types of climate stresses. And then we're using population genomics to try and identify either populations or individuals or species that have genetic variants that might allow them to better withstand potential climate stresses that would be experienced in a warmer and drier world. And this figure is from a different experiment, but what it shows, all these plants right here are the exact same age. The only difference is the type of mycorrhizal treatment they've gotten. And so you can see that there's this great potential for just varying the types of mycorrhizal fungi that plants might be associating with to really create plants that might be more resilient to the future climate stresses that they might encounter. One thing I want to note, though, is that this is a great idea, and I think there's a lot of potential here, but there are still a lot of technical barriers to being able to actualize these organisms as real climate solutions, or climate and sustainability solutions. We don't understand a lot about how to grow them. They're very difficult to work with. And so there's a lot of fundamental knowledge we still need to figure out before we can turn them into practical solutions. So one of the things that's really drawn me to being part of the new school is that it's creating this incredible opportunity and environment to create new collaborations. So we now have a Woods EVP grant that's allowing us to collaborate with Jennifer Brophy over in bioengineering to try and see whether or not we could begin to take the first steps to modify these partnerships to create more resilient plant communities or do things like increase below-ground carbon storage. And so these partnerships are really necessary to take the fundamental understanding that we're building and scale it up towards practical solutions. And so I'm going to end by drawing an analogy. This is a quantum computer. And this is a photo from the Lawrence Berkeley National Lab. And I believe that this is a quantum computer because I've never seen one myself, and most of you have probably never seen one in person. But we all implicitly recognize the value of building a quantum computer and are willing to invest in it. And so if you've taken anything away from this talk today, I hope you appreciate that there's a vast potential for these mycorrhizal symbioses to be leveraged into potential sustainability solutions. We're still a long way to go, but I think it's something that's worth investing in. Thank you.